Hello everyone, I'm very excited to introduce Professor Brian Kennedy to the channel to discuss the latest research of aging. So Professor Kennedy is an internationally recognized researcher for his research on biology of aging and has over 170 publications to his name. From 2010 to 2016, he was the president and CEO of the Buck Institute for Research on Aging and is now the director of the Center of Healthy Aging at the Yong Lin School of Medicine at the National University of Singapore. So Professor Kennedy, welcome to the channel. It's an honor to have
Thanks.
you here.
Happy to be here. Thanks a lot.
Awesome. I'm just hoping if we can start near the beginning of your career, where you were the lead author on a paper in 1997 about Sirtuins and lifespan extension. So are you able to explain to us what the history is of Sirtuin research, and then your opinion about the initial excitement around so-called Sirtuin activators?
Sure. I think that when we first started as a graduate students, and I joined Lenny Guarenti's lab at MIT, and there was another graduate student at the time, Nick Austriaco, who's, by the way, gone on to become a priest after getting his PhD. So that's another discussion. We were trying to figure out what we could do with yeast cells that would be totally crazy and yet still plausible. And so we came up with two ideas, either to try to study aging in yeast or apoptosis in yeast. And so if your listeners don't know, the Saccharomyces is a single-celled yeast. So the idea that it would undergo programmed cell death seemed a bit odd. And so we chose to do aging. No one in Lenny's lab was doing that. So we started the project fresh. Turns out there's a whole field on yeast apoptosis now too, so maybe we would have been okay either way. But at the time we started, it really wasn't to do anything about human aging. There was so much that was unknown about the aging process that we thought looking at a single-celled organism may be an interesting way to at least develop a paradigm for how to think about why an organism ages. And so we set out to find mutants that were long-lived. And so we used this assay called the replicative aging assay in yeast, where you count how many times the mother cell can divide. And when the mother cell divides, it produces a smaller daughter cell, and you can take a needle, a microscopic needle, and remove it, and just count how many times the mother cell divides, and that's its lifespan. So we had to come up with some creative strategies to find long-lived mutants. And through a few good ideas and a little bit of serendipity, we were able to find mutants that were longer lived. And ultimately, what we realized is that they were altering the activity of the sirtuin complex. So in yeast, the main sirtuin that we were interested in is called SIR2 or SIR2. And we were able to show that when you increase the activity of SIR2, it extends the lifespan of yeast. And that was partly initially my work, but also a lot of work by Matt Kaberlein, who joined the lab after I left.
And what was the outcome? Well, what happened after that paper? So that was in
So
97. Then how did the research kind of progress from there?
I progressed by going to do a postdoc and working on cancer for a while. But other people in the lab were trying to understand what sirtuins were doing. And through the work of David Sinclair and Matt and Brad Johnson, it was shown that sirtuins could suppress rDNA recombination and reduce the levels of these extrachromosomal rDNA circles that form in yeast. And that was proposed to be one mechanism by which the yeast cells age. Now, we now know that SIR2 does other things in addition to controlling these extrachromosomal rDNA circles in yeast. So that's not its only role in affecting aging. But it was really exciting at the time because it was one of the first cases where there was a mechanism proposed for how a gene might regulate aging. And then Lenny's lab and David and many others started moving to other organisms like worms to try to see if the orthologs of sirtuins had similar effects on aging. And they also tried to link the SIR2 activation to the lifespan extension by calorie restriction in yeast. So, and that led to, since then there's, 20,000 papers on sirtuins and aging. So it's become a field of its own.
So what's your opinion about the calorie restriction and Sirtuin link? Because from what I gather, reading through the different papers, there's still quite a bit of controversy around that point.
I think that if we answer that, well, we were able to show through some genetic tricks that calorie restriction could still work without Sirtuin there. And then there are other proteins that look like Sirtuin called sirtuins in yeast. And so then the debate became whether they were fulfilling the role of Sirtuin when Sirtuin was absent. And I don't think that's completely been resolved. I think if we fast forward to mammals and look at mice or humans, calorie restriction is causing a constellation of changes. And some of those are mimicked by reducing TOR activity, which we may come to later. Some of those are probably mimicked by activating sirtuins or AMP kinase. And all three of those pathways somewhat talk to each other. And so I don't think that we're going to be able to say at the end of the day that calorie restriction does one thing. It creates a systemic change in cell metabolism that can be partially phenocopied by those pathways. I think that's the best way to say it.
Yeah, makes sense. So with the idea that activating sirtuins is a good idea, what do you think about that statement?
Well, I mean, there's been a lot of, in yeast, if you're a yeast cell, it's a good idea.
Yeah.
There's been a lot of work in worms and flies, which are also somewhat controversial, but maybe we can fast forward to the case in humans, which is what we're probably mostly interested in. There's 7 sirtuins there. Not all of them have been well studied. Some of them are mitochondrial. Sirtuin is considered to be the classic ortholog of yeast Sirtuin, and there have been links between Sirtuin and aging, certainly. Sirtuin has been linked to calorie restriction. That's a mitochondrial Sirtuin. So these enzymes, these proteins are actually enzymes that deacetylate other proteins. So Sirtuin was originally thought mainly to do it to histones, but these sirtuins in mammals have all kinds of different targets. And so in Sirtuin, for instance, in the mitochondria, deacetylates a wide array of mitochondrial proteins. But I think the one that I like the most at the moment is Sirtuin, because the data seems best to me about overexpressing Sirtuin and extending mouse lifespan. There are some links to Sirtuin mutations in long-lived centenarians, which I'm not sure that's published yet, so I won't talk too much about it. And there's now different methods to activate Sirtuin. And what Sirtuin is doing reminds me more of Sirtuin yeast. It's maintaining heterochromatin. It's keeping the genome intact, protecting against DNA damage and activation of retroelements like lines and signs. So I think that functionally Sirtuin may be a closer ortholog of Sirtuin than Sirtuin is. So if I had to bet money right now, I'd bet on Sirtuin as something that really regulates aging.
Yeah, because there was a recent paper that came out about Sirtuin and its potential for extension in mice lifespan, which is very exciting.
Yeah.
Have you had a chance to read that paper?
I'm not sure which one you're referring to.
I think it was published in March of this year, I think. But
you're
overall, I
think
think
you read more literature than I do.
But yeah, overall, I think it's very interesting to follow what's happening with Sirtuin.
It really is, and the ability to suppress these retroelements links the Sirtuin to senescence too, because activation of these retrotransposons is driving innate immunity through sting and C-gas in the cytoplasm when DNA gets in the cytoplasm, and it's also causing DNA damage and contributing to the senescence-associated secretory profile. So linking Sirtuin to suppressing that event is, I think, an important finding in the aging field, because it connects 2 different aging pathways together, which is kind of what we need right now.
So on that point of getting excited about a potential way that we can hopefully extend health span, there's been many different processes that we get excited about that for various different reasons don't quite pan out. So what's the method that we should be using to not get overexcited about, potential dead ends?
Well, I don't know here's the conundrum. I think we've found a lot of ways to extend lifespan in worms and flies and mice and yeast cells. surprisingly, that's a lot easier than we ever expected it would be. I think when I started in the field, there were a lot of people in the field saying you'll never alter aging. It's immutable, too complex. You need to do too many different things. You can't reverse damage. A lot of those arguments. Um, Turns out it's relatively easy. Now, we don't know how much of that's going to extend to humans yet, but there's no reason humans should innately be different than other species, I think. What we've struggled with is trying to understand the mechanisms by which these different interventions extend lifespan. So, it's direct opposition to what people thought in 1990 when I started thinking about aging in the field, which was that we'll probably learn what causes aging, but it'll be really hard to do anything about it. And now I think we're learning it's not that hard to do the stuff about it, but it's really hard to understand what causes aging. And part of it is because it's really a network, in my opinion. It's not just a few pathways that are acting independently. If you look at the pillars or the hallmarks of aging and these papers that came out a few years ago, they identify 7 or 9 tenets of aging, but the important thing about them to me is that they're all connected. And so if you take an intervention that extends lifespan, let's take rapamycin, you can read that as an improvement of each of the hallmarks. And the question is it directly affecting one of them and that's affecting all the others? Or is it more that what we're looking at are different sides of a network or different edges of nodes of a network? And the interventions that extend lifespan are the ones that preserve the network. And so if the network gets preserved, you can read that out in any of the pillars or hallmarks, but that doesn't mean it's directly affecting those. Most of the efforts to try to directly affect things have not been that successful. So, trying to mitigate reactive oxygen species has not really been very promising in terms of extending lifespan. Dealing in DNA damage directly is not so promising, but it's really these systemic regulators that we're targeting that seem to be able to preserve this network that keeps you healthy as you get older. And so that sounds nice, but then when you start thinking more deeply about it, it's really hard to comprehend, I think. And so maybe we really aren't going to understand this without systems biology.
Yeah, it's a very good point about thinking about things at a system level as opposed to just trying to narrow it down to one specific thing. One point before we move on, because I do want to ask you about mTOR and rapamycin. What's your opinion about resveratrol? Because that was one that was going to be the so-called Sirtuin activator that was going to hopefully extend lifespan.
Yeah, I've always been skeptical of resveratrol, and it partly goes all the way back to when I finished my postdoc. I went back to studying aging when I was an assistant professor at University of Washington, and Matt Kaberlein, who I mentioned earlier, had come there as a postdoc. So we decided that let's try to figure out a way. We found a few long-lived mutants in yeast. Let's try to screen the whole yeast genome now and look at all the deletion strains and figure out which ones are long-lived. That turned out to be a 13-year process. but at the start of it, what we wanted to do was just have all of the interventions that we know working in the lab. And so we overexpress Sirtuin, it extends lifespan. We delete FOB1, it extends lifespan, et cetera, et cetera. But we couldn't get resveratrol to work. And it didn't extend lifespan in yeast. And other people have also, so it was initially reported to do that, but we've never been able to repeat that. And then there were people doing biochemistry saying that resveratrol doesn't activate Sirtuin or Sirtu, the human and the yeast enzyme, respectively, against peptides that were acetylated. Now, the original screen that was done was with a acetylated peptide that had a fluorescent moiety on it, and they had a really neat chemistry. The screen was great, had a really neat chemistry that when you deacetylated the peptide, it activated the fluorescent group, and you could score that very easily in a screen. And that worked, but when you took peptides without that fluorescent moiety, it didn't work anymore. And so we published the paper. Another paper got published after that saying the same thing. And then later, David Sinclair's gone back and shown that there are substrates that have bulky amino acids that are similar to that fluorescent moiety in their position relative to the acetyl group. And that for those substrates, Sirtuin activates it. And that our resveratrol would presumably activate Sirtuin toward that. We haven't gone back and followed that up. I think that the other problem with resveratrol is it's a very dirty molecule. It does all kinds of different things in the cell. And so I kind of think most of the field has moved beyond it now. And I'm still skeptical. Other people will tell you their benefits. But the field has moved more toward, NAD and NAD precursors and trying to use those as a means to activate sirtuins and affect aging.
Yeah, that's kind of the conclusion that I've come to by looking at the data and actually the process or the timeline of events with the initial resveratrol excitement and then, research such as yourselves that said, well, hang on, maybe it's not quite that simple. I think the paper that really seals it for me was one that was published last year in 2020 that used CRISPR technology to actually have a look at what resveratrol is actually activating and how is it actually working. And overall, that paper was suggesting that the primary method of how resveratrol works is the induction of low-level replicative stress. So, yeah.
Yeah, which may be true. But this is how science works, right? I mean, you have someone publishes an observation, people try to replicate it. Often it's hard to replicate because there's some condition that's different or some way the experiment's done. And at the end of the day, there may be a few bumps along the way, but you get to more understanding. So I think that it's always the people get nervous when there's controversy in a field, but it's not a bad thing. You need different labs with different viewpoints discussing things and trying to come to the right answer in the end.
Yeah. Absolutely. And you mentioned NAD precursors. So this is something that I've sort of talked about quite a bit on my channel, but I'm very curious to hear from a researcher's perspective. What do you think about the hype around NAD precursors?
Yeah, I'm agnostic at the moment. We've tested both nicotinamide riboside and nicotinamide mononucleotide in our hands, both in normal aging mice, and we've tested nicotinamide riboside in Hutchinson-Gilford progeria mice. The effects are very small, not zero, but very small. And in the progeria mice, we do get a slight lifespan extension, but it's weak. However, having said that, it's clear that NAD levels are declining with aging and the ratio of NAD to NADH. Trying to restore that, I think, has shown in mammalian models that there's some health span benefits to it, if not lifespan. In some cases, lifespans are reported, relatively modest effects. So I'm still waiting to see more human data. I think that one of the challenges, of course, as you know, is you can't easily take NAD directly. So you take a precursor like NR or NMN, and then that has to be converted to NAD, and there's a lot of debate about which of those two molecules is the best. I think that I guess to me the answers aren't all in yet. I want to see what's happening to these biomarkers of aging, and people are starting to do those kinds of studies. I will say that I know a lot of people obviously taking these things, and people report feeling better, more energetic, and while there's certainly a placebo effect to any of these things with aging, there may also be something there. So I'm not negative toward them. I'm still want to see more data, though, I guess.
Absolutely. And you mentioned markers of aging. So one of the big things that's going on at the moment is epigenetic clocks. One of the things that I've kind of always been curious about, and I'm really interested to pick your brain about it, with these epigenetic clocks, do you think that they are a so-called good marker of aging? So what I'm trying to get at is if you find a way, say a lifestyle intervention or a particular supplement that helps reverse the age on these epigenetic clocks, is that a good thing? Or is it just, the chicken and the egg debate?
Well, I look at it this way. The aging interventions came mostly from animal model data. Now we want to test those in humans. The biomarkers came mostly from sort of AI-based analysis of omics data sets, predominantly DNA methylation, but there are others out there too now. Um, and so we have two different things coming together, different areas of research. And so I think if we find that the interventions actually slow down or even reverse these biomarkers, it really is a strong clue that we're on the right track. You know, an ideal aging biomarker is going to predict mortality. It's going to predict functional decline and disease onset, and it's going to be modifiable by interventions. It looks like the DNA methylation clock is meeting those criteria. The third one being still, I think, an open question, but there's starting to be evidence. We know, for instance, in mice that rapamycin and calorie restriction slow the methylation clock. So I think that, and there's some human data. So I think that I'm optimistic about them. I think one of the questions I have is, what does this tell us about DNA methylation? So on the one hand, it seems to me like if you take any deep data set and enough people, you can create an aging clock. It doesn't have to be methylation. I mean, we work with Jackie Han, who can predict biologic age from facial pattern reconstruction, almost as accurately as methylation class. And so is there anything special about DNA methylation, or could you do this from any large data set and by training a program to try to predict age? I think that it's also a fundamental question to me is, what do these clocks really mean, in the sense that a lot of them initially were optimized to chronologic age, and then the error off the chronologic age was considered the biologic age. So if they predicted I'm 54, if it predicted I was 51, then I was 3 years biologically younger than I chronologically am. And now people are trying to optimize that down to 0. But if it gets to 0, we're just predicting chronologic age, which we already know. Now, I think so that I'm not sure you can get there, but it's starting to look like it. And what that means is we're measuring tree rings, not biologic age, you know. And so I think what we have to figure out is how do we match these markers to the true biologic age in the population? So in other words, how do we really measure biologic age and not other factors that may be irrelevant to biologic age? And if you ask me, you know, if you take 10 people at 50 years old, the variation in biologic age may be 5 years. You know, there's probably 2/3 of them fit within somewhere between 45 and 55. But that's a lot of variation, and some of them are outside of that range probably. So we don't really know how to fit these biologic markers to biologic age because we don't know the variation in biologic age. So that's still a challenge, I think. And what we're trying to do is in Singapore, on the academic side, is to really measure the all kinds of different biologic markers of aging, including some of the classical ones, physiologic ones like pulse wave velocity, you know, DEXA scans, things like that. But also to combine them with methylation clock and lipid clocks and or metabolomic clocks, excuse me, and facial pattern clocks. And how do all these things work together? Do they give us a better picture if we combine a lot of them, or are they all telling us similar things? And I think that's an open question as well. So I'm very optimistic about the clocks. I don't think we're all the way to validation, but we're definitely using them, and we're hopeful they're telling us something meaningful. I would say that another thing that excites me right now is, are they just markers of aging, or are they mechanistically telling us something about aging? And so some of the work that Morgan Levine's doing by working backwards from the methylation sites that were identified to predict age and figuring out which methylation sites are predictive of which hallmarks of aging is really exciting to me, because it may begin to tell us why these clocks are predicting aging. And ultimately, we may find that, you know, if you look at one of the Horvath clocks with 350 sites, that these 45 are really measuring, you know, inflammation, and these 45 are measuring stem cell function and something else. And then we'll have a lot better feel for what these clocks mean. So I think trying to deconstruct the clocks mechanistically is really important.
Yeah, I agree, because one of the things that the different companies that offer these clocks to just regular people is how many different sites they are actually sampling.
Yeah.
But you don't know which sites are actually important at this point. So that
You
is
don't.
quite
You don't know
exciting.
how many sites are necessary either. I mean, you can distill those 350 sites in the first Horvath clock down to a few and get almost the same productivity. And that the other thing about these companies is that some of them use just saliva spitting on a piece of paper. Others need blood. Some cost more than others, and they have different levels of variation from chronologic age. But again, we don't know what's ideal for biologic age. So I think people, if they're interested, they should do them. But I really believe for the public, we should educate the public and then let them make informed choices. If they want to
Absolutely.
test interventions, as long as they know where we are, and people are honest about the data they have, I'm fine with them selling and people testing these different interventions, particularly if they're safe, like natural products that are safe at the levels they're being given. And the same with the clocks. I'm interested in where I am, and I think a lot of people are. It's really about informing people accurately about where we are in the research, I think, and then
Yeah,
letting them make their own choices. So.
yeah, absolutely. One of the things that's popped up quite a bit as we've been talking is mTOR and rapamycin. So I'm very curious because there does seem to be this distinction, balance that we're trying to strike with mTOR. That yes, you want to be activating it in certain circumstances, for example, after exercise, but equally you want to not activate it so that things like autophagy can be activated. So what's your opinion about mTOR and how do we strike this balance?
Well, I think you've touched upon the key feature here, in that it's dynamic range. If you make an animal without TOR, it doesn't do very well. It's there for a reason. It's developmental, but also in adulthood. When you have a big meal, nutrients activate TOR, stress activates TOR, TOR is activated in wound healing and immune system activation. And we tend to think that it's good to have things high or low, but the truth is we need things to be high when they're supposed to be high and low when they're supposed to be low. And TOR activity is relatively low in a basal state, and then it gets activated by these stimuli that I mentioned. The problem with aging, I think, is that particularly in stem cells, but in other cell types as well, the turned off state doesn't work anymore. So TOR activation is on almost all the time. And then you have two problems. One is you have TOR on when it's not supposed to be on, so you can't get autophagy and stress responses that you need. But also when you need to activate it, the range of activation is very narrow and you don't get much of a response. So to me, like rapamycin at relatively low dose or some of these other inhibitors, what they're doing is restoring that state. So if you take mice on rapamycin, they still activate TOR after a meal or something. And we did an experiment a long time ago that if you keep rapamycin keep mice on rapamycin for a month, the first day after you give the mice rapamycin, TOR activity is really suppressed. After a month, it's not anymore. The actual protein translation in the cell is identical to what it was before you started. Now, that doesn't mean there aren't specific RNAs that are overexpressed and underexpressed by rapamycin permanently, but what it means is the cell compensates for turning down TOR, and translation goes back to where it was at bulk levels. So, I think it's really the interventions that extend lifespan are really about maintaining the dynamic range of youth, not turning things up and down. So.
And so do you feel that rapamycin will play a part in humans in terms of hopefully extending health span?
To me, it's the gold standard, really, for aging. It works everywhere. The Resveratrol bio data was very promising, I think, and they were using everolimus or a TOR, a different kind of TOR inhibitor, RTB101. But the data was very promising. And, the clinical trial, the phase 3 trial for influenza vaccination didn't work for reasons we can talk about. But I think that the main thing is that a lot of the parameters were improved in these individuals, and they were able to give these drugs to people without any toxicity above background. And these were people 65 and above. So in New Zealand, I think, if I remember correctly. So I think we really do need to explore TOR inhibition because it works in almost everybody's hands. There's almost no debate about whether it extends lifespan. It works in multiple mouse strains, which a lot of interventions don't. And so if we can do it in a safe way, maybe by making derivatives of rapamycin that have the efficacy without the toxicity, then it could be a great model for extending health span.
Do you know if that's being worked on at the moment?
Yeah, there are a number of companies that are doing that. Aovian is one of them. There were two companies spun out by the buck. One was that, and the other is Torcept, which I'm actually involved with, full disclosure. And we're trying to make derivatives of rapamycin that have just that property that we can reduce the toxic side effects. There are other people trying to inhibit TOR through a variety of other ways, including interfering with how amino acids activate TOR and other strategies. So I think that there's a lot of interest in this. The problem is that, clinicians, when they hear about TOR inhibition, they think immune suppression, and they're like, I'm never giving that to a healthy person. And so it's, as a monotherapy, rapamycin is not really that immune suppressive. I mean, unless you overdo it. It actually protects against some kinds of infections and slightly sensitizes to others. It's more immunomodulatory. And so I think that the key thing is dosing it the right way so that you don't get side effects. And so I'm not encouraging people to take it, but I think there are strategies to do that in healthy people won't have long-term side effects. The other thing about rapamycin and TOR inhibition is that, at least in mice, you don't have to do it all the time. You can treat mice for 3 months and stop, and they still live 15% longer, even if they haven't had the drug for a year. So it's almost as if you're resetting an aging clock with it, and then the clock ticks, but the reset effect is still there. We don't know if that's going to happen in humans or not yet, but it does suggest that an intermittent dosing strategy might work in humans, where you maybe took it for a month and then we're off for 6 months and took it for another month, something like that. So I think there are a lot of opportunities to explore TOR inhibition, and really, I think it's still the most likely pathway to modulate human aging at this point.
That sounds really exciting. How far away are these trials from happening in humans where, say for example, the company that you're involved with is actually, doing human trials that are placebo-controlled?
Yeah, we're still at early stage in the Torcept, but Resveratrol was effectively doing that, not for aging directly, but they were doing it for improving vaccine response in the elderly. And so, in a way, the trials are already starting to happen. What we're hoping to do here in Singapore is to test everolimus, which is a rapalog, in a 6-month test, 45 to 65-year-old people that are reasonably healthy, and then use these aging biomarkers. Because what I really want to see is if it modulate these biomarkers of aging. I expect that it will, but nobody's in humans has really shown that clearly yet. So they're starting to happen. I think there's more of a challenge with TOR inhibition just because of this perception, and natural products are moving a bit faster right now. But I'm not, I don't think we should be against the idea of giving drugs to people, even if don't have some classic disease indication. I think if we wait until people are sick, we're doing sick care at that point, and it's not very effective. It costs a fortune, and we can see that it's not working as well as it should. We should be doing healthcare, which is how to optimize people's health and protect them before they get sick. And that's really a life course approach, I think. So I think if we're taking people in their 40s and 50s before they get sick, identifying their risk of aging rapidly and targeting that with drugs, I think that's a very healthy way to go forward. And I'd like to see that being validated and employed in a more widespread use to extend health span.
Absolutely. Yeah, sounds really, really cool. And cottoned onto the mTOR, TOR complex is AMPK. So I personally take metformin on days where I don't exercise. And that's just based on a few observational trials in humans. What's your opinion about metformin?
I think it's also promising to test. The animal data is not as good as rapamycin, to be honest with you.
Yeah.
There is some hints of benefit, but it's not a big effect. Um, these retrospective studies showing that people with diabetes taking metformin have surprisingly low mortality rates in their 60s and 70s, lower than people that don't have diabetes, is a striking finding. And retrospective studies are fraught with all kinds of problems. Like, I'll give you an example. If you get diagnosed with diabetes, you're going to the doctor's office a lot. If you're healthy in your 60s, you're probably not going at all. And so you catch that cancer at stage 2 or 3, and whereas in a otherwise healthy person, they may not go to the doctor and it may not be caught till stage 4. So that there are all kinds of complications with these studies, but that human data does look really promising. And I think that trying to figure out what metformin is really doing is another challenge. It does result in AMP kinase activation, but probably not directly. And it's probably a mitochondrial electron transport chain inhibitor, but it's also been, every year there's a new paper saying, now we know what metformin does, and it's always something else. So the drug's been used for 40 years and we still don't know mechanistically what's going on. There's another natural product from Ayurveda called berberine. And in our hands, berberine and metformin have very similar effects even in yeast cells. And so, it may very well be that berberine has a similar mechanism to metformin, and that's something that can be taken as a natural product. I think it's another very intriguing pathway, and it certainly links both TOR and metformin link metabolism to aging. And that we already know there's a big intersection there between altered metabolism and accelerated aging. So I'm positive about metformin. I think the challenge is, and I'm very supportive of the TAME study that Nir Barzilai is promoting. The challenge is that's a hugely expensive study. It's a 3-year study, tens of millions of dollars. Nobody wants to pay for it because metformin costs 4 cents a day and it's off patent. Metformin was chosen because it has a very good safety profile, and I understand that. But we can't afford to do that for every intervention that might affect aging. And so that's why we've decided to take smaller studies, shorter term, and use biomarkers rather than disease prevention. Because if you're trying to prevent diseases, you need lots and lots of people and long-term studies, and those are extremely expensive. So I hope TAME works and it really encourages the FDA to rethink health span. I think that's important. I just don't know if that route is the best way to find the interventions that have the biggest effect.
The final question around TOR that I want to ask you is fasting. So in mice, it appears that so-called time-restricted feeding or intermittent fasting is a great option. The issue that I've always had with those trials is that if a mouse fasts for 16 hours, that's completely different to a human fasting for 16 hours. So I personally do a fast of 7 days every 3 months, but I'm very curious to hear what your opinions are about fasting.
Well, first of all, I think it very likely works. I mean, a lot of this came from calorie restriction, and it's sort of in 2 different ways. One is people realize that when you calorie restrict mice, so normally mice have food in the cage all the time. They eat when they want. It's like sitting in front of the TV with the refrigerator right there. You can get food anytime you want. And when you calorie restrict them, especially when they're multiply housed, so 3 or 4 mice per cage, you put 60% of the food they would normally eat in. And when you put it in, they eat it. And then it's gone. And then there's a 12-hour window before you put more food in. And so essentially, you're not just reducing the calories of the mice, you're fasting the mice. And then people decided to do what's called every other day feeding to mice. And they found that on the days the mice have food, they eat almost twice as much as they normally would. So the total food intake is almost the same as what the mice are eating in an ad lib diet, but they're still getting a lot of the benefits. And that really pointed to fasting as a way that might underlie some of the benefits even of calorie restriction. And then if you look at the human situation, nobody wants to do calorie restriction. It's very hard. not nobody. There are people that do it. I'll get the Calorie Restriction Society mad at me, but it's not something that's going to undergo widespread adoption, I think. Trying to eat 1,400 calories a day and still get all the right balance of macronutrients and all the micronutrients is hard to do. It's hard to do because you have to be really disciplined, but it's also hard to do because you have to know what foods to eat to get everything you need. And in the mice, we have these pellets that are, have the ideal amounts of each food. So if we give them a very low calorie content, we still know they're not malnourished. And I'm not sure that's happening when people are, making semi-random choices and trying to eat very low calories. But fasting is much easier for people. So what you do, there's 5-2 diet where you eat normally 5 days a week and fast 2 days. There's time restricted feeding, 12-hour, eating all your food within 12-hour window or within 8-hour window. I know people doing it in a 4-hour window. And then there are companies like El Nutra, which I'm involved with, full disclosure again, and they make diets that mimic fasting. This is research that came from Walter Longlow's lab at USC. And part of the reason they developed those is that even though they did fasting studies and showed medical benefits in different disease contexts, people didn't want to comply with the fasting requirements. So now that you have boxes of food that keep certain pathways like IGF-1 down and mimic the fasting state while still giving you something to eat. And people are very excited about this approach, and it's selling well. So I don't know which one works the best. There's certainly arguments to be made for various different fasting approaches. There may be some risk associated with too much time restricted feeding, and so it's still a lot to be learned. But almost any of these strategies are showing benefits right now, and in some cases, like medical benefits too, not just with things like diabetes, but with reducing side effects of chemotherapy, with affecting autoimmune disease. So I think it's a very promising area of research. And certainly calorie restriction or fasting, both are going to turn down mTOR activation, among other things. So that there's probably overlap there as well.
So on that point of different diets and different macronutrients, the keto diet's huge at the moment.
Yeah.
I've always been a little bit worried about the keto diet, and I'm very curious to hear from your perspective, who's really stuck into the research in mice and in humans. What's your opinion about the ideal macronutrient balance?
Well, I try to tell people to keep a good balance of macronutrients. Don't try to overdo one and cut another one out completely. I mean, certainly if you cut carbohydrates out and eat a lot of fats, you lose weight. I mean, because your body's not adapted to deal with that diet, and so you don't maximize energy from the diet, and you lose weight. Whether that's sustainable and whether that's healthy in the long term, which is what we're looking for aging, is entirely another debate. I think that if you look at Steve Simpson's work on mice and a lot of work on other species as well, if you keep animals at an isocaloric diet, so in other words, all the animals are getting the same calories, and then you switch the macronutrients around, what you find is that it's high carb, low protein that leads to longest lifespan, not high protein, no low carb. And I think we tend to say carbs are bad for a couple of reasons. One is we're eating way too many calories in general. And carbs are a great way to eat way too many calories, especially processed sugars, which I would say should be minimized. Complex carbs, I think, are pretty healthy, but processed sugars, not so much. And so if you want to overeat, Twinkies and cupcakes are good ways to get there. And so that carbs are bad in that sense. And then going back to what I just said, our bodies are not used to massive influxes of processed sucrose and which immediately gets converted into the body and activates insulin production and alters metabolism. I think if you're eating your carbohydrates from fruits where they're more complex, it takes a while for your body to break down those carbohydrates, and it takes a while for glucose to accumulate. So that's a much healthier approach. So I think keto is not bad. It may be good, but it depends on how you get there. And if you get there by fasting, which is a great way to get to keto, by the way, it's probably very healthy for you. But if you're constantly unbalancing your macronutrient load, I'm more concerned about the long-term effects of that. So.
Yeah. Thanks for that insight. The next thing that I want to ask you is a paper that you co-authored in 2020 about alpha-ketoglutarate, which seemed to show lifespan extension in female mice, but across the board it seemed to extend health span. So yeah, I'm really interested to pick your brain about this trial and about alpha-ketoglutarate.
Yeah. First of all, I don't think there's a huge gender difference here. There was a trend toward extension in males, and if you put 4 cohorts we've done together, you actually get statistical significance. Now, of course, if you do that, you try to publish a paper, the reviewers will, tell you that's not the right thing to do, which is, I understand. But I do think there's a small benefit in males as well. So we started this treatment at 18 months and with not that high of a dose of alpha-ketoglutarate. And so we got about, like I said, around a 10% extension in lifespan, but a 50% reduction in frailty. So we want to argue that this compound is compressing morbidity in mice. It's keeping them healthy very late, and then they kind of crash at the end. That would be optimal if it worked that way in humans. Of course, we're trying to test that. And this came out of a lot of research. So a company called Ponce de Leon Health worked with Gordon Lithgow and I at the Bach. And first thing we did is take a lot of natural products that were reported to affect aging in one way or another, and we tested them in worms, took the ones that worked and started combining them together. The idea is to find combinations that have effects on aging, because you can get IP around combinations of natural products, but not single ones. And also to sort of survey the landscape to see what's going on. And so by far our biggest hit was alpha-ketoglutarate out of that. There are other things that we saw benefits from, and the product that Rejuvent that they sell contains alpha-ketoglutarate and vitamin A for men and alpha-ketoglutarate and vitamin D for women. So, and that's because we saw beneficial effects on frailty by combining the two. And I can come back to vitamins if you're interested to. I think I'm very interested in those in aging. But AKG seems to have a robust effect. We're now doing controlled clinical study. I'm part of this company too. Ponce de Leon Health is sponsoring controlled clinical studies at Indiana University, and we should have data back on that in the near future. And that's with the product. And here at NUS, we're going to test just alpha-ketoglutarate because I want to get a better handle on mechanism and not have the complication of the other ingredients. One of the things we found is that the sustained release version that we've generated probably works better than ones that AKG that's not sustained release, because it gets through the gut and slowly releases AKG throughout the day. It has a lot of effects. It's sort of like, Sirtuins and mTOR, and that if you start looking at pillars of aging, you see benefits. And it's a lot like NAD in the sense that it participates in hundreds of reactions in the cell. And so it's not an obvious place to figure out which reactions are important. So, with NAD, yeah, it activates sirtuins, but it activates a lot of enzymes, and it could be others that are linked to aging too. AKG is the same. So both these metabolites go down, and you're trying to add them back. So we're kind of beating our heads against the wall trying to understand the mechanisms of action of alpha-ketoglutarate right now. We have a lot of people that have taken the product and done methylation tests with a company called Trumi Labs, and they've shown that they have a reduction in biologic age. We're trying to publish this paper after 6 months of taking the products. This is obviously not a placebo-controlled study. people paying money to buy the product, and so they're highly motivated to see an effect. It has, those kinds of issues, but it's very promising, and we hope we see something similar in the controlled clinical studies.
Awesome. Because one of the questions that I'm very curious about, when I first read this paper, I think it was sometime last year, it was talking about how much alpha-ketoglutarate was being given to these mice. And from what I can gather, it was 2% of their diet, which to me sounds quite a lot.
Yeah.
If you try and extrapolate that to a human diet.
Yeah.
So
Yeah, and I think we had a limited ability to test different doses. And so one of the things we're doing in mice is going back and testing different doses. I'll say that the AKG, you can take a lot of it in human studies without toxicity. So there have been a lot of clinical studies on this molecule. Bodybuilders have been pouring it in their bodies for a long time. Endurance athletes use it. There was a study on osteoporosis where I believe they went to 10 grams a day in humans, which is like, lunch. And and they showed benefits from postmenopausal loss of bone density, but they also saw no toxicity in 6 months of that. So I think that this is a very safe molecule, and it's not. So what's in the product is actually maybe lower than it should be. And so one of the things we want to go back and is do higher levels of testing in humans when we do the study here at NUS. I think that I tried doubling the dose and that's recommended, and I'm not dead yet, so that's a good sign. So but we really don't know what the appropriate dose for AKG is. But we didn't want to start with a very high level in humans because even though there's a lot of good safety data on it, we wanted to be careful first. And this study we're doing at IU is ostensibly it's a safety study, even though none of the ingredients we have in the product they all have very good safety profiles at the dose we're recommending. But we want to validate that first and then go forward.
When do you think those trials would be published so that we can read them?
Hopefully in the next 6 to 9 months. I mean, it got pandemic. I think we should I've made pandemic into a verb at this point. Yeah. Because, we just started the trial and everything had to shut down. And actually, I'm still hopeful we'll see some really good data out of it, but it's a bit scary because if you look at the control parameters for people that are the placebo control, they're all over the map right now because you had people that were working, they weren't working, the restaurants were closed, they were locked down, they were sick. So you can see people in some of the clinical markers we're using that they were sick, so it's not going to be the best, I think, not just for ours, but for any studies right now. It's not going to be the best time to have done clinical trials. I think we're going to see a lot of squiggly lines on the control side. So hopefully we'll still see some benefits, and we're going to be able to really analyze that data in the near future, but it always takes time to put it together and publish. So it's, but I think that I'm still optimistic that this is going to do something interesting.
Awesome. No, that sounds really exciting. I can't wait for this to come out. You mentioned vitamins.
Yeah.
Yeah. Are you able to expand on that? What are you excited about? Because when I've gone through Cochrane reviews and meta-analyses, it seems that a lot of, I wasn't that excited about a lot of vitamins. There are a few that I take that I am excited about, but I'm very curious to hear your opinion.
None of them work in the meta-analysis. That's what you don't like, right?
Essentially, well, there's a couple that I'm hopeful for. For example, vitamin D, vitamin K2. Those are interesting to me in terms of their health span benefits. But
I think we can throw out all these studies that were done because what did they do in most of them? They didn't measure and optimize vitamins in the body. They just gave some people a high dose of a particular vitamin and somebody else, another group didn't get that, and then they didn't show much benefit. The truth, though, is that you really want to optimize the level of these things, and too much could be just as bad as too little. We know that's true with vitamin A, for instance. It can be toxic when it gets too high. And so what we're trying to do first in mice now is do personalized longevity studies where we try to optimize the levels of the vitamins in each mouse. So we really want to try to personalize the dose of these vitamins and see if we can have an effect. So we know already with vitamin A that we've tried multiple lifespan experiments. The first time we tried too high of a dose, and we confirmed that indeed it's toxic. So the mice didn't live as long. We went back and tried a similar dose, but then we stopped. We started at 18 months and stopped around 22 months. And at that point, 20% of the mice had the toxicity. The other 80% lived 20% longer than the controls. And so that tells us that if we get it right, we can really have a big effect on aging in these, and at least in this animal model. So now we're trying to take blood every month and reconfigure the dose so that we keep, efficacy markers where we want them and toxicity markers where we want them in each animal. And I'm betting we get a big effect on aging. And by extrapolation, I would bet you if we took 10 vitamins and did that for individuals every 2 months, we might have a big effect on their aging too. So I think these vitamins are really important. It's just that the studies weren't done in a way that actually measured, usually weren't done in a way that actually measured the levels of these vitamins in the bloodstream and what the effects were. So it was too much of a blunt instrument. If we take a personalized approach, I think there'll be real benefits.
Yeah, I hear what you're saying, but I suppose the counter to that I've got in my mind is if you've got a really good diet so it's a wide and varied diet, it's got lots of vitamins and minerals in it, is there then going to be an advantage to supplementing a particular vitamin?
Well, so what you would do is measure at baseline, and if people don't need things, don't give it to them. So yeah.
So you're saying just individualize it to...
Yeah. I'm not saying everybody needs vitamins. I'm saying we need to optimize the levels. And but I do think that what we would call optimal is a narrower range than what the FDA calls. I just had a blood analysis done in my vitamin B12. It was considered in the normal range, but I would say it's low. And so, I need to do some supplementation there. And but you're right. If people come in and they have all the right levels of everything, the last thing you want to do is give them a bunch more.
Yeah, because that's what I sort of do with my patients in the clinic. If they come to me, usually we're doing a blood test every 6 months or 12 months anyway, and on that I'll add B12, folate, iron, all that other magnesium, just making sure that it's roughly yeah.
And I can't emphasize that enough. By the way, if people are interested in their health, they should be testing these things. They should be getting clinical panels done and looking at what the levels of all these risk factors are for various diseases and trying to optimize now. Don't just assume that since you don't have a disease, you don't need to come in and figure things out. Like my homocysteine's high. I didn't even know that. So, that's something that I probably should be doing something about.
Final question. So we've talked about alpha-ketoglutarate, we've talked about NAD precursors, and we've talked about rapamycin and mTOR. Are there any other molecules that you're particularly excited about?
Yeah, I think there's a bunch of them that are coming down the path. And at the end of the day, we don't even know which ones are going to have the biggest effect. And we're not even talking about like stem cell therapies and gene therapies and other things that might be even further away, but have bigger effects in the long run. Spermidine, I think, is one to pay attention to. It's an autophagy activator, has other properties like everything else. Guido Kramer, Frank Medeo have done a lot of work on it, and it was shown that it extends lifespan in mice, and we've been able to repeat that in our labs. That's one of the things we do is test a lot of different natural products to see what repeats in our hands. We were able to also show that it improves metabolism, particularly in mice eating too many calories, which again would align with about 90% of humans. So that's definitely an intriguing one to look at. I'm interested by urolithin A, which is identified as a mitophagy activator. We've started studying that in the lab. That's Johan Ower's work and other people. And there's also activators of SIRT6 that are starting to come on the market. And I think that as I said earlier, SIRT6 is an exciting molecule. So if you can really activate it, that may be a beneficial effect on aging as well. And, that we could make a list of 50 things if we tried hard enough, drugs and natural products. We're working on a drug called Gemfibrozil, which we haven't even published yet. This is a fibrate that's used for hypertriglyceridemia, and we find interesting effects on aging, probably through inhibiting amino acid uptake. So I'm of the opinion now that drug, the target that was identified, may not even be right. So we're going back and doing a lot of biochemistry right now. But as I said earlier, I don't think it's going to be that difficult to slow aging. I mean, we tend to have this idea that complexity means difficulty in intervention. But if you look at it a different way, if you think of it from that network approach, where you're trying to still maintaining homeostasis in your body, you're still healthy, you're still functional, maintaining that equilibrium is a whole lot easier than waiting till you spin out of equilibrium and get some disease and trying to reverse entropy. And right now we're spending all our money trying to reverse entropy and finding out it's not so easy. And if the aging interventions are more about maintaining homeostasis, and in my opinion, that's why it's turning out that it's a lot easier to do that. And I think a lot of it will be translatable to humans as well. That's speculation, but that's my guess right now. So, even though it's a complex process, that doesn't mean it can't be modified. And so I think that I'm very excited. I think there's going to be a lot of strategies that will slow aging. And the next challenge is going to be figuring out which ones work best in which people. And we really don't have a good handle on that right now, but it's certainly going to be somewhat personalized. And that's probably the phase 2 of the human intervention studies.
Well, I think that's a really positive way to finish the podcast. So thank you so much for coming on the show. I'm really hopeful to get you back one day so we can go into a bit more detail about those molecules that you've mentioned. But I can't thank you enough for your time. I'm sure the audience is going to really appreciate it.
Great. I'm happy to do it. Thanks a lot.